Okay, everyone, here's take number 3 with this video, how I want to go through our central dogma with you. I had a few issues before, but I think I got it all figured out. This is going to be a video that's associated to your assignment. I'm going through transcription and translation coding process and also identifying mutations. So we'll do this together, but I wanted to make sure that I had this option so you can pause and revisit this if you have any specific questions as you're working through. Um, the first thing I want to talk about is this over here on the right side. This is called an amino acid codon chart. So I'm just going to call it a codon chart. But it's going to be giving us the information of our messenger RNA strand and that code, and then tell us which associated protein is going to be brought along the way for translation. So on the outside here, I'm going to just do a quick highlight. These are our amino acid names. There's 20 different ones, and some of them have multiple codes that are associated to them, which is what we're kind of seeing here when their ending code has multiple options, right? And the reason for that is it allows us to have some unique genetic codes to them. Each individual can have a unique genetic code, but still be able to create the same amino acid or protein strand, but still have some uniqueness to them, which is really, really cool. And if we happen to have, a point mutation or a base error that's happening, it's still highly likely that we might end up with the same amino acid that we intended, despite that mutation, which is another cool kind of check and balance piece that our genetic material has. So those are the protein names on the outside. We just shorten them with 3 letters when we go to code and write it out. Some specific ones we should be aware of over here in green. This is MET. This is our start codon. And the MET, I'm going to write start. We should be familiar with that one. And how do we know that's our start codon? It's going to be based off of the messenger RNA code. I'm going to start inside out when I figure this one, but it's going to go A, U, G. And that's going to code for start. And that should be found on every beginning of our messenger RNA transcripts when we do this coding process. So that's kind of how you would go through and find those. There are a couple more over here in red that say TER. Those are our termination sequences or our stop sequences or stop codons. So there's 3 different ones that are associated to that. I'm going to write stop. And you can write MET or TER as your amino acid, or you can write start and stop, whatever floats your boat on that. I'll show you what that looks like as well. But again, our 3 options for that would be UGA, UAA, or UAG. Those are our 3 stop options. So that's how you would identify those. All right, I'm going to erase these highlights so that later on we can go through those together and not be confused as to what I'm writing. So then let's see. I want to talk about our central dogma piece again, just kind of recapping that. Remember how we can go from utilizing DNA as our master code for our genetic material, and that can go through and do its semi-conservative self-replication process for mitosis. It can go through transcription and create some RNA transcripts that can then leave the nucleus if we're talking about eukaryotic cells, and meet up at the ribosome and then perform this protein synthesis, amino acid combination, which is highly important for the survival of all organisms, right? And this is the backbone of biology. So that's our breakdown. Remember, DNA is going to be made up of A's, T's, C's, and G's. RNA is going to be made up of AU's, C's, and G's. And then our amino acid is going to utilize that codon chart. So for your assignment today, you're going to be given this original DNA templates sequence that I've just highlighted towards the top. We're going to be working off of that and comparing that strand to some mutated DNA sequences later on, and we're going to figure out, what type of mutation is associated with it. Before we get going on this, I just want to kind of recap. If I were to ask you to go through and use this original template strand right here and perform DNA replication, remember those bases are going to be read 3 at a time. That's kind of called a codon or our reading frame. This original template strand is like the one that is used for everything. And then it'll make a complementary strand or it'll make a messenger RNA strand, depending on whether we're doing replication or transcription. But it's always going to be the one that we code off of so that we have that master one where there isn't any specific mutations that could go with it. Right? So when we do our complementary base strand for DNA, which I'm going to just write above it, we have A's, T's, C's, and G's, and they're matching up with their opposite partners, right? So instead, this would look like A, T, G if I'm going through DNA replication, right? T, G, G, A, C, C, G, C, T, G, C, T, G, A. And you can call me out later on if I made any errors. But that is what our DNA strand would look like if it was going through and doing its semi-conservative replication. So then these two strands would go off together and live in harmony, right? So there's that one. But we want to go through and write out our messenger RNA transcript. And when we do that, right, we're going to be using the opposite or the complementary bases. But this time our A's are going to turn into U's. Our uracil is found in our RNA. And there are no T's involved here. So make sure you take your time as you're doing this. And we're going to code it 3 bases at a time, right? So this time I'm going to have A, U, G. That should be ringing a bell for some of us. U, G, G. A, C. C, G, C. U, G, C. U, G, A. Okay. So next up, I'm going to go through and take this messenger RNA transcript and use my codon chart on the right to figure out what my amino acid sequences are. So I'm going to start with my first codon here, AUG, and we talked about this one being an important one that we should be aware of, right? So I'm going to start on the inside and work my way out. AUG codes for methionine, which is the same thing as a start codon. So I'm going to write MET. You could also write start in that area above if you want to, but you only need the 3 letters as you're going. All right, so I'm going to grab a different color and do some highlighting for UGG. UGG codes for tryptophan. So I'm going to write TRP underneath that one. So the amino acid that is associated to a tRNA molecule for UGG is called tryptophan, and that's going to be pulled in and brought during translation. Okay, my next one, AAC. I'm going to go AAC, which is going to code for aspartanine. ASN. I'm going to keep it going. Do just a few more highlighting so we're on the same page. C, G, C. I'm going to make sure I wrote that one right. Codes for arginine. ARG. UGC. Codes for cysteine. And then my last one is UGA. And if I do that one, UGA codes for termination or stop. So I can write TER for termination, or I could write stop in that location, right? So now I've gone through and done my coding for my original strand. Now I'm going to take that information and I'm going to be comparing that to the mutated strand that I have here. So my first step is I'm going to identify where the issue is in my genetic code. So I'm just going to compare each thing. I have TAC down at the bottom, which also aligns with the TAC with my original strand. I have ATC, which does not align with the next one, ACC. So I know my error is over here at the T, and I'm going to try and make that one look a little bit prettier with my circle. Be my friend. Okay, my T is the issue where it should have been a C up top. So I know that's my one piece. And if I look at the rest of the strand, it matches up. So I'm just kind of keeping that as a note for later on. Then I'm going to go ahead and code my messenger RNA transcript, just like I did before. A's pair with code for the T's that were there. The A's go to U's, G's, and C's, right? Oops. Try not to do that. U, and now I have an A instead and a G, and then everything else should still be the same as I code it up. You can confirm that I've done this correctly. U, G, A. Okay, and then I'm going to go to my codon wheel or my codon chart and then do the same thing and make sure I have all the different pieces aligned and I have everything correctly written out. So my first one, which we talked about before, oh yeah, they keep the circles, which is really nice. The first one that I talked about before is AUG, right? And AUG should be found at the beginning of all of our codes because that is our start codon, right? AUG. That's a good thing, right? So there's my methionine. And I always like to write start. Sorry to change it up from before. It's the same as MET though, right? And then my next one is going to be UAG. So I'm going to go UAG. I ran into a termination sequence or stop codon here. And when I hit a stop codon, when I'm writing out my amino acid sequence, I do exactly that. I stop and I don't code for anything beyond that point because that's how translation works with our ribosomes and our messenger RNA molecules. So I'm done with my coding for amino acids. I'm going to just ignore everything after that. And now I'm going to figure out what type of mutation I have. Was it a substitution and just a point substitution where there is a base that was switched out, or it could be multiple bases that were switched out in that location, or was it a frame shift mutation where a base was added or deleted, right? So we already went through and did that confirmation piece of the fact that there was originally a C cytosine and now there is a T and everything else is the exact same. So we know that this is going to be a point substitution. Okay. And then I need to figure out, was it a silent mutation? So that means, how did it affect the amino acid sequence that I wrote? Silent means that even though I had a base substitution and a switch, I still ended up with the same amino acid. I'm just going to write that note on there, amino acid. Let's get into that. That's not the case here, though, right? We originally had tryptophan, and this time I have a stop codon. So nope. Missense means that it would have coded for a different amino acid, diff, and then I always like to code amino acid as AA, so that's what's going on there. And then nonsense mutation happens when we encounter an early stop codon, and it causes confusion for the entire reading frame. Early stop. So hopefully that's pretty clear that we just encountered ourselves an early stop codon. So we have a nonsense mutation. All right. And then a recap. If we were to have identified a frame shift mutation or an insertion or deletion, you can usually identify those right away because the coding strand here for a mutated DNA sequence is going to be either elongated because of the insertion or abbreviated or shortened because of the deletion. When that happens, they call it a frame shift because when you add something, right, it's going to completely move everything down the line and shift all the remaining amino acids to be different. And the same thing when you delete one base, it's going to like delete something in one of your assays. It's going to shift everything forward where it shouldn't have been in the first place. So oftentimes that leads to the fact that your ending sequence never encounters a stop signal and it messes up all that genetic material. So there wouldn't have been like a UGA at the end. It could be a deletion or an addition of multiple bases so that we have just a whole brand new amino acid that was added in there. So, 3 bases were added in that might cause that to increase or be deleted. Or it might just be 1 or 2 bases that were added and deleted, and that would cause like this shift forward and backwards. Like I talked about how all those amino acids are affected, but you still encounter a stop signal. So that's how this would be different from that one. There's still a stop, but over on the left here, there wouldn't be one. So you're going to take your time and go through that coding process for the rest of the mutated DNA sequences and then identify which type of mutation you have encountered. Yay, that one worked. Have a great day.